Hello everyone, so in this set of notes for Microbio from my class, we're going to be looking at the host-microbe interactions. So we're going to look at pathogenesis and then also the relationships between different microbes and us, because they're not all bad ones. Not all bacteria can cause disease. There are a lot of beneficial bacteria. Some of them we'll be going over today, as well as the history of host microbe interaction as well. So, with that being said, a little bit of history. So, back in the day, like thousands of years, people thought that diseases were divine punishment or caused by bad air. It wasn't until Leeuwenhoek's discovering microorganisms in the 17th century until people started to think that these microbes were the root cause of many of the diseases instead of punishment or bad air. It would be his discoveries kind of putting it into the mindset that there might be this causative effect of the microbes being present everywhere, and some of them are causing disease. And then later on in 1876, this is when we have Robert Koch. And when he does his studies, he offers the evidence for the germ theory of disease. Alright, so let's talk about the main players in the microbiota, or in these host microbe interactions. That would be, of course, the bacteria. Bacteria are ubiquitous. This means that we're going to always be in contact with them daily. They're going to be breathed in. They're going to be on surfaces, etc. And most will do no harm. Usually people think that all microbes are pathogens, so all of them are bad. No, this is not the case. Most of the microbes are actually harmless. Some of them will be colonizing the body surfaces, others will just be pushed off or slothed off. Most of them that are ingested are attacked in the stomach, or they're removed through the feces. So we have ways of removing some of these bacteria. And then pathogens are again the microbes that will cause disease in healthy individuals. Again, not all microbes are pathogens. We have three main interactions with microbes. First would be mutualism. And this is a relationship where both partners benefit, where the bacteria will make vitamins in the large intestine, and us, the hosts, will provide warmth as well as food. There's also commensalism. This is another type of relationship. This is known as a positive neutral relationship, because one of these organisms will be benefiting and the other one is just not harmed in any way. And then our last interaction would be, of course, parasitism. And parasitism, as in other biology classes, this is where one of the organisms is gaining and then the other one is losing something. It's benefiting at the expense of another. All pathogens are parasites. Even prokaryotic ones. Alright, let's talk about the normal microbiota. So, we've discussed before that there are a bunch of these microbes that just routinely reside on the body's surfaces and don't pose any threat to us. That's why they're called the normal microbiota. And we have both the resident and the transient. The resident microbes are going to be there for extended periods. Whereas the transient ones are going to be temporary. When we have this normal microbiota, we're going to have colonization. This is the process of them getting into our systems and staying on our surfaces. This will begin at birth. And where it begins in birth would be from breastfeeding as well as formula. The composition of whatever's in the breast milk or in the formula, that will determine what species are going to colonize your normal microbiota. Again, the composition will be different among individuals and over time. So you have a specific normal microbiota specifically for you, and then someone else has a different one. There's going to be changes with the physiological state and the lifestyle of the host. For example, there's more firmicutes in people who are obese, while there are more bacteroidetes in those who are thin or underweight. Again, that just shows the composition will be dependent on the individual's lifestyle as well as physiological condition. They are going to also protect against the harmful species of microbes. They're going to cover binding sites, use up resources, and we're also going to see some of these normal microbiota species make their own toxins to stop others. When you suppress your normal microbiota by adding antibiotics, you could accidentally cause the colonialization of pathogens. So if antibiotics are misused, we could have the inhibition of lactobacillus, So lactobacillus is out, and then Candida albicans will grow, and then you get candidiasis. There's one example, and then again, Candida albicans is a pathogen. Oral antibiotics could stop the intestinal microbiota, and the Clostridium bacteria could increase in the intestine if that normal microbiota is gone. Alright. Again, they have a protective role. They're going to stimulate, sorry about that, not simulate. They're going to stimulate the adaptive immune system. Again, the adaptive immune system, this is when we have the T cells as well as the antibodies from the B cells. So the A, B's that bind the microbiota also work for pathogens, or I should say on pathogens. So, for example, mice in the microbe-free environment have underdeveloped mucosal-associated lymphoid tissue, or MALT, because they don't have that stimuli from the normal microbiota, so therefore they can't use these malts. There's going to also be oral tolerance. So the immune system will decrease the response to the microbes on gut and as well as the ones on food. And so from this principle, we have something called the hygiene hypothesis. The hygiene hypothesis states the insufficient exposure will lead to allergies. Remember, allergies are because of these haptens, or these usually normal particles are thought to be malignant, or they're thought to be pathogenic. So that's why the immune system goes into whack when that ever happens. And then they also help digestion. For example, there's a species of bacteria that helps us digest beans. Let me pull it up here. Let me go breakdown of fibers, specifically in beans. Normally we cannot break it down. However, when we have those bacteria in our gut, there's a specific strain of bacteria that live there. They're able to break down the fibers in beans, so that's why we have so much gas production like nitrogen and CH4 methane. And from those bacteria breaking it down, they create those byproducts, and that's why we're gassy after eating beans. So think of that whenever you have Taco Bell next. That's the reason why. Those little microbes are actually helping you by breaking down the fibers. Alright, let's talk about the principles of infectious disease now. Keep making that mistake. There we go. Alright, our first principle, we've already talked about this, colonization. In regard to the pathogenic bacteria, this is when the pathogen establishes itself and multiplies. Infection is to refer to the pathogen. There could be subclinical infections This means no symptom. However, there are also infectious disease. And when we talk about infectious disease, this is where there are very drastic symptoms. Symptoms are going to be the subjective effects experienced by the patient. And what we mean by that would be pain or nausea. Someone can't tell if you're nauseous or not. They aren't you experiencing your first person life, so they can't experience that pain with you. They can't tell. However, signs are much different. Signs are objective evidence. That even other outside sources could see, such as a rash or swelling. We also have primary and secondary infections. Let's go here. So a primary infection would be the initial one. This is the initial. However, the secondary infection is from the exposure to another pathogen or the same pathogen due to the damage from that primary infection. Right, so now we've talked about primary and secondary infection. Let's talk about primary pathogens. Or this whole section is also on pathogen virulence, so that's kind of like the potency or their ability to make you sick. So primary pathogens. These are going to cause disease in otherwise healthy individuals. Such as plague, malaria, measles, and the flu. Get your flu shots if you haven't yet, please. You want to stay ahead of these things, especially if it's a primary pathogen. You need to get that flu shot, especially in this season when I'm making this video. As well as your COVID, I know they updated the boosters recently. Please also do that. Okay, so we also have opportunistic pathogens, or opportunists. These cause disease only when the body's innate or adaptive defenses are compromised. So, disease only when immune system, and it could be either innate or adaptive, is compromised. Or if they're in the wrong spot or wrong area. So, for example, would be Pseudomonas. Then we also have, again, virulence, actually. Sorry, I'm pronouncing it strangely there. This is going to be the pathogenicity. And then virulence factors. It's another vocab word. Factors will allow microorganisms to cause disease. So, contagious diseases again. I think that's self-explanatory what that word means. And the infectious dose, which is also known as the ID50. So this is like the lethal dose for substances. The infectious dose is the number of microbes needed for infection. And we're going to infect 50% of these cells. For example, shigellosis, ID50 is about 10 to 100 of those bacteria versus salmonella, needs as many as 10 to the 6, not 16 to the 6, 10 to the 6 ingested salmonella enterica in order to be infections, in order to cause infection. Sorry about that, really fumbled the words there. Alright, now let's talk about the duration of symptoms. These are going to be kind of self-explanatory. We have acute duration. This is going to be quick symptoms and short term, I should say. Like strep throat. We're going to also have chronic. Chronic means that it's going to be slow and it's going to be long term, such as tuberculosis. And then latent. When we talk about a latent infection, this is going to never be eliminated. And it's going to be in the host tissues without symptoms. However, when we decrease our immune system, this could cause it to reemerge or flare up. For example, chickenpox. This virus will actually hide in the nerves, and it's going to then later produce these particles to cause shingles. And then there's also TB, cold sores, and genital herpes as well. There's going to also be different ways to talk about the distribution of a pathogen. These are also kind of self-explanatory, localized. It's only in one area. And this is going to be like a boil caused by Staphylococcus aureus. A systemic infection will go throughout the body, such as measles. And emia, when we talk about emia, that means in blood. This means that the pathogen is in the blood. So we have things such as bacteremia, which is going to be bacteria circulating in the blood. Not necessarily a disease state. This could actually be caused by toothbrushing. Toxemia, viremia, and then sepsis. This one is very important. Sepsis is when the life-threatening inflammation, I'm going to have to actually highlight this one, caused by infectious agents, is in the bloodstream. So when that inflammation moves into the bloodstream, that's when we're going to refer to it as sepsis. Alright, let's talk about Koch postulates. Right, so he's going to use these to establish that anthrax will cause anthrax, the disease. So number one, the microbe must be present in every case. I'm going to just put here anthrax. That's the disease that he originally studied. Number two, the organism must be in a pure culture. From the disease host. Number three, the same disease must be produced when pure culture is introduced into another host. And number four of the postulates would be the organism must be recovered from all the infected hosts and must be the same. So the mechanisms of pathogenesis, how these microbes could cause a disease that would be monitored by Koch postulates. Sometimes there are toxins that are ingested. Sometimes they go to the mucous membranes. Sometimes they go into the tissues to produce toxins or to avoid defenses. And the pathogens and hosts will be playing this balancing act, where the hosts and the pathogens will kind of evolve to balance each other out. Alrighty, let's talk about establishing infection. So the first step would be adherence. Again, this is attachment via adhesins. It's a protein to the receptor. Bacteria are going to be using fimbriae or pili. Or a capsule. There's extremely specific binding to exploit the host cell receptor here. Alright, next step, step 2 would be colonization. This is where the bacteria could cause a biofilm. We have bacteria known as siderophores. So these will bind iron. Again, by binding iron, we can't use it. Nor can our normal microbiota, so they're able to outcompete. There's going to be hiding from secretory IgA. Again, this is just one of the antibody classes. We're going to hide by rapid pili turnover, antigenic variations in IgA proteases. So protease to directly attack that IgA antibody. There's also pili turnover. And then there's also antigenic variation from all these mutations and different strains popping out. And again, we're going to have competition. And the microbes are going to be shooting these toxins at the normal microbiota, trying to compete with them. Right, we could now establish infection. And some bacteria actually do this via something called the injectosome. And it actually looks like a syringe. If you look up injectosome online, you'll actually see an electron microscope image of it, and it looks like a syringe. It's insane. We're going to deliver effector proteins. And this is going to be in gram-negative bacteria. This is going to also be known as a type 3 secretion system. This is going to allow for the uptake of the bacteria. Injectosome, again, it looks kind of like a syringe. If you look at an actual electron microscope of it, it looks like that. It's going to shoot those effector proteins right into the host cell. Right, now let's talk about invasion. They're going to be breaching the barriers, these pesky pathogens. So, skin penetration. It's a difficult barrier to penetrate, first off, so the bacteria will use cuts and injuries. For example, Staphylococcus aureus can enter wounds and cause infections that way. Also, Yersinia pestis is injected by fleas on rats. When they are trying to suck blood off these rats, they actually just create that injection, and then the bacteria goes into the rats. So fleas on rats, fleas on rats. Okay, penetrating the mucous membranes is actually different. A lot of penetration here of different kinds. Entry point for most. And this is going to be directed uptake by the cells. So the pathogen will actually induce the endocytosis. For example, Salmonella will do this process called ruffling. Oh, I meant to highlight it. There we go. This is going to, again, like shake and scramble that membrane to gain entry into the host cell. They could also avoid host defenses. They could hide to avoid all these things I'm about to list. So, complement proteins. They could also hide from phagocytes and antibodies. Shigella and Listeria monocytogenes. These are going to actually direct transfer from intestinal epithelial cell to adjacent cells by causing host actin polymerization. And this is going to kind of like propel them. It's going to shoot them into direction. Again, that's from actin polymerization. Okay, so how do cells avoid destruction from the phagocytes? So we're going to be using a C5a peptidase to degrade the chemoattractant of C5a, as the name suggests. This is going to be found in Streptococcus pyogenes. And we could also have membrane damaging toxins. These are going to just kill cells. Straight up kill them. And then the strep pyogenes will make something called streptolysin O. So they both have a peptidase for chemoattractants, and then they also have membrane toxins like streptolysin O, Streptococcus pyogenes. They could also use capsules. Capsule in bacteria, this will interfere with opsonization. And then Streptococcus again, so some will bind host regulatory proteins that activate C3b. Again, another Streptococcus, but the pneumonia one. There could also be the M protein. Cell wall of Streptococcus pyogenes will bind to the regulatory protein that inactivates C3b. And then we also have FC receptors. A ton of fancy kinds of methods. These are going to bind the FC region of antibodies. So it's going to prevent those antibodies from working. Okay, so surviving within phagocytes. Some bacteria can survive inside those phagocytes. So there's escape. One of the methods that they have is just to straight up escape. Listeria is going to produce a molecule that forms pores. And then through those pores, they're just going to make their dastardly dash out of there. Prevention of the phagosome-lysosome fusion. A species that will be doing this would be Salmonella. And they're able to sense ingestion by macrophage and they block via a protein. And then there's also survival. One of them that can actually survive, this is extremely rare, is Coxiella burnetii. And this is Q fever. This is the rarest method of surviving a phagocyte. Serum-resistant bacteria are going to resist complement proteins. For example, gonorrhea, the bacteria that causes gonorrhea. will bind to the regulatory proteins to prevent the formation of that membrane attack complex. Remember, that's after one of the pathways in the complement system has made that convertase, and then we get the splitting, and then some of it will become that membrane attack complex, which just pokes holes into the unwanted cell. But gonorrhea bacteria are going to be able to bind to that regulatory protein to avoid this MAC from being put together. And they could also avoid antibodies. IgA protease is going to be the cleaving of IgA found in mucus. This is going to again be made by gonorrhea. And then there's also the mimicry of host molecules. So they're just playing like dress up. They're going to cover the surface with similar molecules, and therefore they appear to be part of the self. They're actors. Streptococcus pyogenes again, so S. pyogenes is a jack of all trades. Most of these are going to be S. pyogenes methods. They're going to use something called hyaluronic acid. A polysaccharide found in the tissues in order to play dress up here. And again, when these pathogens infect you, they're either going to be direct, so toxins produced, or indirect damage, which is the immune response. Sometimes the damage may help the pathogen to exit and spread. Right now we're going to be talking about exo versus endotoxins. Endotoxins are going to only be from gram-negatives, and they include the lipopolysaccharides of the outer membrane, specifically the lipid A. That's going to be the main one. Exotoxins are going to be both gram-positive and gram-negative, and they're usually proteins. Let's talk about the exotoxins in much more detail, since we have a ton more of those first, and then we'll go into the endoproteins. Okay, secreted. So these exotoxins are leaking or secreted into tissue after the bacteria are lysed. Foodborne intoxication or foodborne illness. Most are denatured by heating. They could be local or systemic. And then, so since they are proteins, these exotoxins, we could generate antibodies against them. That's a sigh of relief there. However, many of them are fatal before the immune response. So, kind of a double-edged sword there, a little bit. So vaccines are going to be critical. We're going to use toxoids. inactivated toxins in the vaccine, or an antitoxin, which is the neutralizing antibodies. Should also mention it's a serum. Okay, exotoxin types. They're going to be enterotoxins, which are for the intestine, neurotoxins, which are going to damage the nervous system, and cytotoxins. The cytotoxins are going to lyse cells, hemolysins are a specific category as well. These are going to attack the red blood cells and lyse those instead. Some of them will insert into membranes and form pores. And again, Streptococcus pyogenes with streptolysin O. Again, we just saw streptolysin O up here. They're also going to use it to kill cells, so that's a way they have hemolysis. There's also phospholipases. And phospholipases are going to attack the membrane. The alpha toxin of Clostridium, perfringens, will cause something called gangrene. Usually exotoxins will have this A, or active subunit, and this B, binding subunit. So the active subunit is the toxin itself, and the binding subunit will again bind, and it's going to determine the cell type to be infected. All exotoxins have that. A super antigen, this is a type of exotoxin, I think. I don't really know for sure because the placing of the slide, it's kind of smack dab in the middle of this section, but let's go over it. Super antigens are going to bind the MHC class 2 and the T cell together tightly. So when this happens, the T cell freaks out and interprets it as antigen recognition. And then the toxic effect is for massive cytokine release. So when this mistake is created, the TH cells are going to release a ton of cytokines. This is going to cause toxic shock syndrome toxins. So TSST. And then, of course, this is going to be caused by staph and strep. But guess which species of strep? Streptococcus pyogenes. Again. Other exotoxins include exfoliatin from Staphylococcus aureus. So here we have something called exfoliatin. And this is going to cause scalded skin. It's going to destroy the binding of the layers of the skin. And the bacteria is growing in one small area, but the toxin spreads out. Then there are also some hydrolytic enzymes too that could also help. spread the bacteria. Okay, endotoxin. Again, this is only going to be in gram-negative. And it's the lipid A of the lipopolysaccharide on the outer membrane. This is going to cause the inflammatory response. When it's localized, this is going to help clear infection. However, when it's systemic, this is going to be septic shock because of that widespread response. This is dangerous. There's going to also be toll-like receptors, or TLRs, that are going to induce cytokine production. And the T-independent antigen response of B cells at high concentration. And so I'm just going to draw a ton of arrows because that thing is going to be firing off like crazy. Peptidoglycans can also trigger fever, sepsis, and septic shock. And we're almost finished. Okay, damaging effects of the immune response. So inflammation again. They could cause some damage. They could have the phagocytes release enzymes, and that could really mess up our own cells. Adaptive immunity. Has this form of damage. We have immune complexes. When you have the immune complex, the antigen-antibody complexes can form. They settle in the kidneys and joints and activate the complement system, leading to inflammation. Then complement, and then inflam. And that's, again, throat infections of strep pyrogenes. We could also have cross-reactions or cross-reactive antibodies. And these are going to go out and bind to the body's own tissues. So this is going to be the autoimmune response, which we've talked about in a previous video. An example would be rheumatic fever from, again, Streptococcus pyrogenes. Viral pathogenesis. So now let's talk about viruses really quickly at the end here. There's going to be, of course, that stage of binding and invasion via the specific receptors. Again, they need to hijack the cell, but in order to hijack the cell, you need to bind to it and then insert the genome, the package. Right, so they're tricks. They're very tricky, as we saw with all those COVID variants. They avoid all these different things. They avoid interferons. They block the expression of genes or activations of enzymes. They could also avoid antibodies. They could go create cell fusion known as syncytium, actually. Sorry about that. Very strange word. They could modify the surface antigens, and they could outpace the antibody production. That's why we are in an arms race with microbes, especially viruses. We saw with the pandemic, we need to be on top of these things because we could have all these different variants all coming at us at a single time if we're not careful in how we're selecting a vaccine and we're not making good lifestyle choices. Then that's just going to select for more and more difficult variants to treat with either bacteria or even viruses. Because again, we're in an arms race, a constant arms race with these pathogens. They're always going to be one step ahead because of all their mutations. I just need to make sure we keep an eye on all that. Another way that they could avoid the immune system would be the RNA virus replicases. There's also the HIV reverse transcriptase, which again introduces so many mutations because the lack of proofreading. And then when those get translated, those RNAs get translated, then you have variants of the virus, as we saw with COVID again. Alright, thanks so much for tuning in. This was a long video, but we got through all the chapters so far for this next exam that's going to be on Tuesday for me. Alright, thank you so much for tuning in. Please have a good rest of the day and do something nice for someone.